Good morning, guys. Can you hear me?
Yes.
Yes.
Thank you. So, Monday, November 9th. We only have less than a month, right, in this class. Basically, just, 8 lectures, I think? 8 lectures and a few more exams, right? A few more exams. And 2 chapters to go through, but we did not finish lipids last time. And so I think I would like to take the whole day today to finish the lipids, and we will start the membrane transport on Wednesday. That's why I did not send you the new slides. I will send you those for Wednesday. And there's still quite a bit we need to talk about in terms of lipids. And so one whole lecture would be appropriate, I thought. Okay, so let's get to it. Share screen. All right. So we started to talk about sphingolipids last time. Sphingolipids. And I think it will be good for us to go back to this slide. To this slide. Can you see this slide? Can you see these slides? Hello?
Yeah.
Yes?
Yes.
Okay. So sphingolipids, again, the main difference is that the backbone will not be glycerol. We'll just see in a moment. And they have a long chain amino alcohol, sphingosine. Okay. So specifically on the next slide, we saw this last time. I can see these red arrows, which means we already talked about this, right? So sphingosine, and you told me that this was orange. I remember that. So in orange color, we have sphingosine. So here we have it looks like it's glycerol, but it's not quite glycerol, right? So there are 2 hydroxy groups like in glycerol, positions 1 and 3. But on position 2, we have an amino group. So that would be sphingosine, and sphingosine also has a long fatty tail, right, with a double bond in it. And double bond is positioned in a allylic, at a allylic distance from this hydroxy group. So that's sphingosine. Now, if you add this fatty acid, make an amide on the nitrogen, without the head group. So the head group, still nothing, still missing. So this will be ceramide. A ceramide, so that's the full backbone of this sphingolipid without the head group. Now, once you add the head group, so for example, phosphocholine, so remember this is choline, right? I think by now you already remember the structure of choline. Part of the neurotransmitter, acetylcholine. And phosphate, don't forget the phosphate, and don't forget that this is diester, right? Phosphodiester. There's oxygen on this side, oxygen on this side, and there's oxygen on that side. So it's a phosphodiester. This will be sphingomyelin. So sphingomyelin is the most common representative of a sphingolipid. Now you can have carbohydrates. So sphingolipids are famous that head groups can have carbohydrates. So for example, glucose could be one of them. So this will be glucosyl cerebroside, right? It's going to be without the phosphate. So this will be neutral. It will be neutral glycolipid. Now you can have a di, tri, or tetrasaccharide, in which case it will be called a globoside. Or I can have a complex oligosaccharide, and we'll talk about those. There are different kinds of those. And specifically, one particular should be a sialic acid. So we'll look at the structure of that. If you have a sialic acid, that means it's a ganglioside. Ganglioside. And this in ganglioside, there's specific nomenclature. We don't really need to be concerned with nomenclature of gangliosides in this class. But if you have one sialic acid, then the letter here is M. And depending on the structure, depending on where sialic acid is attached, there are several types of gangliosides. There's GM1, GM2, GM3. But again, this is beyond our class. We really don't need to worry about it. Okay, so these are the structures of sphingolipids. Let's see what else on the slide that we need. Phosphodiester linkage, we talked about that. Okay, sugar continuous glycosphingolipids are found largely in the outer face of plasma membranes. It may make sense, right? The polar head groups. So, and so when they say outer face, remember I was telling you the cell membranes have the outer face and they have an inner face, right? So the outer face of the cell plasma membrane will face the extracellular medium, right? Remember I was telling you that if this is the cell membrane, like so. And so the glycosphingolipid, so the sphingolipid, let's say it's this one, right? So let's fill this in, fill this circle in. So this will be outer. Outer, inner. So sphingolipid shown here. Let's say GM2, ganglioside with the sialic acid will be on the outer surface. All right. So the most common one is actually sphingomyelin, and that's with the phosphocholine. So we just saw the structure, and here is the structure again with stereochemistry. So it's important to realize there are actually two stereocenters in sphingomyelin, right? And so sphingomyelin, it's a particularly one of the major functions of this would be in the formation of myelin sheath that surround nerve cells in animals, for example, in central nervous system, CNS, and peripheral nervous system, right? That surrounds the spine, for example. Sorry, that is in the spine, right? Brain, spine. So so nerve cells, they're insulated. Just like think about electrical wire. Electrical wire needs insulation, right? So insulation, obviously you don't want to touch the electrical wire. That's part of the reason why there's insulation. And second reason is so that it's protected from any kind of external magnetic electric fields, and so the signal, so the electric pulse is transmitted more rapidly. So that's another reason why there's insulation. And the same thing in biological systems, our nervous system is actually insulated. And there are types of cells known as glial cells. Glial cells. Glial cells, which important to know for a number of reasons. In the brain, glial cells give rise to the type of cells known as oligodendrocytes. Dendrocytes. Now, when you take anatomy or physiology, you should probably study this. Oligodendrocytes, and so these are glial cells. And in the peripheral nervous systems, they give rise to types of cells known as Schwann. Schwann cells. So, so these kind of cells, oligodendrocytes and Schwann cells, they produce a lot of this lipid. They produce a lot of this lipid, and so the Schwann cells and oligodendrocytes, they associated with the neurons, and they basically produce this sheath, the myelin sheath that will surround the neurons, and there will be lots of sphingomyelin as well produced by these types of cells. So unfortunately, these kind of cells are also kind of can be an Achilles heel of especially our susceptibility to cancer. So for example, glial cells, they give rise to the most fatal type of cancer known as glioblastoma. Glioblastoma. Some of you, I'm sure, have heard of this kind of cancer. Fatal brain cancer. Not a single patient has survived glioblastoma yet to date. We cannot treat this. Highly invasive, basically invades all parts of the brain, and it comes from glial cells. So they have an important function, but they can give rise to the most horrible type of cancer there is. Actually, this is something that we do in my research group. We work on some natural products that have activity against glioblastoma cells. Specifically, these glioblastoma cells, the reason why they're hard to kill is because they are resistant. You already know this word. What's the most common type of programmed cell death is called? Programmed cell. Say it again.
Apoptosis.
Apoptosis. So apoptosis actually, there is a reason why it's called a programmed cell death, right? So there's specific signal transduction pathway that is associated with apoptosis. And so glioblastoma cells have defects in this machinery, in apoptotic machinery. And so they cannot die through apoptosis. And so majority of chemotherapy induces apoptosis in cancer cells, but the chemotherapy does not induce apoptosis in glioblastoma cells. And so the compounds we work with in the lab, they do not induce apoptosis. They actually induce a different type of cell death known as paroptosis. And I actually have a grant from the National Institutes of Health, paroptosis, that actually is aimed at developing compounds which induce this mode of cell death in glioblastoma cells. And hopefully glioblastoma cells will be susceptible to this, and we can successfully treat brain cancer. So see, it's all related, right? It's all related. Biological systems, there's just like we talk about a lipid, right? A common lipid sphingomyelin, and you can see just through a short discussion, we're talking about the deadliest brain cancer there is, or the deadliest type of cancer there is. So, all right. Any questions? So that's the sphingomyelin. So Yes, so some associations for you. So for example, sphingomyelin, I told you this looks like glycerol, but it's not glycerol. But if there was molecular glycerol, that's a common phospholipid, right? Known as phosphatidylcholine. Phosphatidylcholine. I don't know, some people may pronounce this phosphatidylcholine, but I've heard it pronounced both ways. I prefer phosphatidylcholine. And you can see, so glycerol, but the phosphocholine part is the same. So the head group is the same. All right, so these glycosphingolipids are responsible for the difference in our blood types, right? So you can see that these lipids, so who would have thought, right? So it's the lipids on the surface of red blood cells, which dictate which blood type we have, or the blood group. And and specifically the sugar content of these glycosphingolipids. And sugar content is regulated by enzymes known as glycosyl transferases, right? So these enzymes, they can add they can make glycosyl bonds, or they can break glycosyl bonds, right? That's why I call transferases. And if there is no glycosyl transferase to mediate, to add anything additional to the carbohydrate tail of the sphingolipid, so these people will have the what's known as O antigen. So let's look at the picture is much better actually. So let's say, so this is glycosphingolipids, which will determine the blood groups. So this will be in a cell membrane of the red blood cells. So we have ceramide, right? Ceramide, which is sphingosine and a fatty acid attached to the nitrogen. That's the amide. So we know that. And so here is the carbohydrate tail. So carbohydrate tail of this, so there's a table here. So it starts with glucose, then yellow galactose, blue square N-acetylglucosamine. This kind of circles is galactose again, and then the red fucose, red triangle. If there's nothing else, this is O antigen. Or antigen. And so remember what antigen, when we talked about antibodies, anybody remembers what antigen is? Antigen is the site on a particular molecule to which antibodies are developed, right? So if you have a foreign object, this entire object can be the antigen, or you can have most of this object to be recognized by as your own, but there'll be some small part of it which will be foreign. And then this small part, which is foreign, which is not present in the host organism, that will be referred to as an antigen. And to this antigen, we will develop antibodies, okay? So people with the blood type O will basically, since there's nothing else, will have zero antigens. There's no antigen here. People with the A blood type, they will have additional sugar here, N-acetylgalactosamine, shown here. Okay, people with the B antigen will have additional galactose. So we'll have a nothing, N-acetylgalactosamine, and galactose. So remember these three situations: nothing, galactose, galactosamine. Okay, and so these three situations will give rise to 4 different blood types, blood groups. So So let's say let's stay with this for a while, for another minute or so. So if there is nothing, if there's no antigen here, there'll be no antibodies. No antibodies will react against the O antigen. No antibody. If there is no antibody that will react to this antigen, then this person with this blood group, who can this person give blood to?
Everybody?
Anybody, because there's no antibody against O antigen, there is no galactose, right? Now, A antigen, now here we have N-acetyl galactosamine, right? So here, so there is already antibody, right? And there is a antibody which is specifically reactive against A antigen, A antibody. And so for people, for example, who have blood type B, right? Or people who have blood type O, they have this A antibody because they don't, because this A does not exist in their body. If the A does not exist in their body, they have antibodies against it. And so they will react against it and may result in the immune response, right? So same for B antigen. For B antigen, you can have a B antibody that will be cross-reactive with that. B antibody. And so this table is a very short scheme that can be used, I guess, by medical professionals, right? So these are 4 blood types. So again, let's start with the end. Let's say group O, right? So you have no antigens. You have no antigens. Why? You have no antigens because you do not have galactosamine, N-acetyl galactosamine, and you do not have galactose. You have no antigens here. No antigens. And so if you have no antigens, you can give your blood to anybody. But you have in the blood, you have antibodies against A antigen and against B antigen, which means if you have this blood type, you cannot accept blood from anybody other than people with the same blood type, right? So for A and B, right? So if you have blood type A, you have A antigen, but you have B and anti-B antibodies. So a person with a B blood type cannot give you his blood because this person, sorry, these two cannot be mixed, right? Because if you have this antigen and you try to give the blood to person with a B, then this person has antibodies against you, and that can lead to immune response in a person with a blood type B, and vice versa. And AB are sort of the luckiest of all of them, of all of us, right? They have antigens against anything. So which means that nobody can take their blood except for people with this kind of blood type, right? And so, but they can accept blood from anybody because they have no antibodies. They have no antibodies. And so they can accept blood from A, B, and O, and there'll be no immune reactivity. Do you guys have any questions? And so the antibodies, the particular type of antibodies, you know, there are different kinds of antibodies. So these ones are IgM antibodies, which are present in serum. Do you guys have any questions about how this works? So you can see it's a sphingolipids present in the red blood cells, and specifically the particular kind of carbohydrates which are attached to the ceramide, right? which attach to ceramide, which will dictate what blood type you have and can be the reason for immune reaction. All right, let's keep cruising. So the lipids can be degraded. In fact, they will be degraded, right? So they're synthesized and degraded all the time, and they're continuously recycled. And so, for example, so these are phospholipids. So there are different kinds of phospholipases. So remember, lipase is an enzyme that breaks the esters of fatty acids. And so, and if you have phospholipid, the enzyme that breaks the fatty acid ester will be called phospholipase. And depending on which ester is broken, there are different designations for that. So for example, this is phospholipase A1 that breaks the terminal ester. Phospholipase A2 breaks the middle ester. And then you have phosphodiester. There are two types of esters here. And there is phospholipase C and D, depending on which bond is broken, right? There are two of them. And so gangliosides, for example, are degraded via a series of enzymatic cleavages as well. And so failure to correct gangliosides results in the buildup of lipids in lysosomes. And in fact, there are a bunch of diseases associated with incorrect processing of gangliosides. So I don't really want you to memorize this, but it's important to know that for each biological molecule, there is a host of enzymes which are involved in synthesis and which are involved in degradation of these biological molecules when they're not needed anymore, or there's excess of those, right? So, so for example, this one, this ganglioside here. Now, I think, so new 5-acid, this is the sialic acid. This is the sialic acid, right? This is the sialic acid. It's actually attached here. So this is ganglioside M1. And so you can see, for example, this galactosidase. So remember, the ace means breaks, cleaves something. And that the prefix beta galactose means it cleaves beta galactose residues from something. And so if this terminal sugar is galactose attached to the rest of the oligosaccharides through beta linkage, so the enzyme that cleaves this is known as beta galactosidase. And if this enzyme is underexpressed, there's shortage of this enzyme, there's a mutation in this enzyme, then that will lead to the disease known as generalized gangliosidosis. Gangliosidosis. Now ceramide, now you can see here, hexoaminidase A. So I guess this one takes off the N-acetyl. What's this? Oh, there we go. And N-acetyl galactosamine. So if this cannot be taken off, this leads to another disease, Tay-Sachs disease. So there are a whole bunch of other diseases depending on what defect in recycling there is. So this is just for general information. As I mentioned to you, I don't really want you to memorize, but I want you to see how these are degraded. One sugar at a time, right? And there is an enzyme responsible for each sugar. All right, so we will talk a little bit about then other types of lipids, which are not based on glycerol or sphingosine anymore. So steroids based on sterol rinse system, So I briefly mentioned that to you some time ago, right? So you have this 6665 type ring system, typical of a steroidal backbone, right? So if this is particular one is cholesterol, this molecule is cholesterol. What's important to know about the structure of cholesterol? Most of it is obviously hydrocarbon. There is a small polar head group, which is not charged, right? So it's pretty small. So cholesterol primarily is hydrophobic and dissolved in cell membrane. And cell membranes, it modulates fluidity. I already asked you this question. You told me that it makes the membranes more rigid, right? So it thickens the plasma membrane. So this actually quite a could be counterintuitive. Like if you didn't know that, if somebody asks you, so let's say you have plasma membrane, right? Like so. So one way to look at it would be, let's say you have the very nice fatty acids, and you put cholesterol in there. Put cholesterol. And then you get something like that, where you have to create space. Here's your cholesterol. Like that. So which will break up the interaction between the fatty acids, correct? So like if you didn't know about the rigidity, you would think that actually cholesterol should make the membranes more fluid.
Well, doesn't it depend on temperature? So like in hot temperatures where the membrane would be more fluid, the cholesterol helps keep everything together. But in really cold temperatures, when the hydrophobic tails would get more rigid, the cholesterol helps keep it fluid while it's cold, but when it's hot, it helps keep it more or firm.
Oh, I didn't know about that. I didn't know anything about it. So we're talking about the biological, physiological temperature of 37 degrees Celsius, right? Unless we're talking about organisms that either cold-blooded or thermophilic or something like that, right? So, but also have to keep in mind that cholesterol is primarily present in cell membranes of animals, right? So cholesterol, for example, is not present in bacterial membranes. So, so I understand the argument about temperature, which is a good one.
That's just what I remember from physiology.
Yeah.
Yeah.
Okay. All right. Thank you. Yeah. So the way I kind of understand this is, the fatty acids, again, many of them are unsaturated, correct? So this will be the ideal scenario when they all lined up properly and so on and so forth. But in the real cell membrane, there are holes. There are empty spaces, right? There are empty spaces in cell membrane that needs to be packed, that needs to be taken up by something. It's almost like, you pack in a suitcase, right? And you have an empty spot in the middle, and which makes the suitcase susceptible to, you press on it and it gives away, right? But if you pack that little spot with a pair of underwear, that is always useful to bring extra, right? So then it rigidifies it, right? So the same kind of thing with the cell membrane, I would think. That's cholesterol basically looks for these empty spaces in cell membrane and finds home in those and rigidifies cell membrane. Does this make sense to you? Yeah.
Yes.
Anyway, so I gave you my secret knowledge of cholesterol, so don't tell anybody. The understanding why cholesterol works to rigidify cell membrane. All right. So we mentioned that you can get it from food, you can synthesize. Right, so it's bound to protein in the bloodstream. We can measure it, right? So low density, high density lipoproteins. And so it's the ratio of the two. Remember when you measure cholesterol in the blood, right? So low density are the bad ones. So those ones tend to deposit cholesterol in and clog arteries. Okay, so many hormones are derivatives of sterols, of cholesterol, and specifically they're oxidized, right? So cholesterol by itself only has one oxygen, right? But if you add more oxygens, there are other derivatives. So for example, testosterone, which is produced in the male reproductive system, in the testes, right? So is a male sex hormone. Estradiol is a female sex hormone. Cortisol, you can see here lots of hydroxy groups, but the same 6,6,6,5 backbone, right? So this one is responsible for glucose metabolism. Now aldosterone. Anybody knows what this is responsible for? So this is known as a mineralocorticoid. It's responsible for sodium regulation in the kidney, right? So for example, one way for us to keep our blood pressure under control is the retention or release of sodium by the kidney, right? So the more sodium you have in the bloodstream, the higher the blood pressure. And so if your blood pressure drops, the kidneys will start retaining sodium in the bloodstream, which will raise the blood pressure. And so aldosterone and cortisol, they're both produced by the adrenal gland. So this is involved in regulation of blood pressure. Now prednisolone. So these are inflammatory related to inflammation steroids. This one I don't remember. It's very unusual. All right, so there is biologically active lipids that do some chemistry. So for example, this lipid that comes from arachidonic acid. So this is a 20 carbon fatty acid, also present in the lipids in cell membrane. There are 4 double bonds, all cis. So this molecule actually undergoes series of enzymatic transformations. And specifically, it's a precursor for types of molecules known as prostaglandins. And prostaglandins are famous for the regulation of inflammation and fever, right? So the way fever works in the body, the way it your temperature rises is through the initial reactions performed on one of the lipids, one of the fatty acids in cell membranes, arachidonic acid that goes to prostaglandins. These have receptors. And also not just the inflammation and fever, also pain. For that reason, for example, non-steroidal anti-inflammatory drug, NSAIDs, right? So aspirin, ibuprofen, Tylenol, all these block the production of prostaglandins and block the pain response, for example, right? So also other types of molecules are produced from arachidonic acid, thromboxane. This is responsible for the formation of blood clots, right? There's what's not shown here is prostacyclin. So thromboxane and prostacyclin work opposite of each other. So thromboxane causes the formation of blood clots, whereas prostacyclin actually thins the blood. And so it's the balance of the two which will decide if you bleed or you have blood clots. right? So very tiny balance, but you can see also it's also all comes from lipids. So lipids are not only structural stuff. The lipids are important precursors for other biologically active molecules. Leukotrienes are important for smooth muscle contraction in lungs. For example, if you have asthma, you're producing too much leukotriene, and that production needs to be blocked, and you take medication for that. All right, so this is the famous process for the production of vitamin D. Vitamin D, we know we need sunlight, right? We need sunlight for that. There's actually a series of 4 reactions. So it starts from this dehydrocholesterol, and there's a beautiful reaction that is not studied in OChem2, but it's studied in more advanced classes. So there's this bond 10-9, which can be broken, and specifically broken using light. Without light, it will not undergo cleavage. And this type of reaction is known as electrocyclic ring opening. Electrocyclic ring opening. That gives another step. There will be double bond migration that gives rise to this ring open structure, and then a couple of hydroxylations in other parts. So if this occurs in the skin, right, that's where UV light can reach. UV light will not be able to penetrate anywhere beyond the skin. So this will occur in the skin. Then this goes in the liver. In the liver, it will undergo hydroxylation position C25. Then it goes to the kidney. In the kidney, it will undergo hydroxylation position C1. And that's you get vitamin D. And vitamin D is responsible for what? What's the main function for that?
Bone strength.
Say it again.
Strength of bones.
Yeah. So how do the bones they get stronger?
Calcium and phosphate uptake.
That's right, calcium. So vitamin D is involved in the regulation of calcium in the body. So one of the main function of that is to increase absorption of calcium in the intestine, right? So calcium from food will be absorbed in the intestine into the body, into the bloodstream, and vitamin D is an important molecule in that process. Now, vitamin A, so that molecule is responsible for vision. So it starts with beta carotene, this undergoes cleavage. And now this is something that actually I did not know. I remember there was one lecture. So I know this material starting from retinal and retinol. And in one of the lectures, I was talking about retinal and retinol and their relationship basically to vitamin A, right? And somebody said beta carotene. I said, no. Beta carotene is not involved in the process, but I was actually wrong. It is involved. It's not vitamin A by itself, but it's a precursor. It's a precursor that can undergo cleavage. And so, so here we have this highly conjugated molecule. This is all trans retinal. Retinal is an aldehyde, right? Al stands for aldehyde. And there's all that stands for alcohol, retinol, all trans. So this is the vitamin A1. And so all these double bonds are trans. And in the body, what will happen specifically in the retina, this will undergo oxidation and isomerization to cis. So this double bond here, 11-12, will undergo from isomerization from trans to cis, and the alcohol will undergo oxidation to the aldehyde. So this is known as 11-cis retinal, and which actually binds to a protein known as rhodopsin. Rhodopsin, so a main protein responsible for the perception of light, right? And it specifically has 11-cis retinal bound to it. So this conjugated system, you can see 1, 2, 3, 4, 5, 6. This will absorb light in a visible region, right? In a visible region, it will absorb light, and the energy which is absorbed will be used to force this isomerization from cis to trans. And now imagine, so cis and trans have different geometries, right? So the way it's drawn is kind of, they look the same, but you remember that if you have a cis double bond, then you have this geometry, right? If you have trans double bond, then you have this geometry. And now imagine if this is part of the protein of rhodopsin, right? And you go from cis to trans, then obviously this will have a major impact on how this molecule binds to the protein, right? How this molecule binds to the protein. And so the conformation of the protein will change, and that will initiate the nerve signal transduction pathway. And so the light, the photon of light will be converted to an action potential in the new yeah, I don't specifically remember how the structure of the eye and how the retina are specifically related to the ocular system. So there's optical nerve. Anybody knows how the optical nerve, how it is linked to the retina, and how the signal is transduced from one to the other?
The optic nerve is in the back, and it's just ganglia nerves. So they have photoreceptors that relay information to it.
Mm-hmm.
So the retinal is just in the retina and in the photoreceptors, and it absorbs the light and sends it to the optic nerve.
Okay. Which then puts together all the processes the information from multiple inputs, right?
Right.
Okay. All right. So let's see what else we have. I think we're almost done. I estimated this perfectly. Now there's other kinds of vitamins. Also, you can see the highly hydrophobic. So there's antioxidant vitamin E. There's blood clotting cofactor, phyloquinone. Some of them have double bonds, others don't. Warfarin, a blood anticoagulant. Some of the important lipids also present in the mitochondria, for example. So this is coenzyme Q. So you can see this is an electron carrier. Whenever you have the quinone type system, quinone, hydroquinone. Now you were not born yet when there was photography, when the photography was based on using films, photographic films. But the photographic films were based on this redox process when you have a quinone. This receives 2 electrons, receives 2 protons, and undergoes transformation to hydroquinone. So these types of molecules are involved in redox processes and very popular in biological systems. So quinone to hydroquinone, And so these types of molecules are involved in electron transfer. Electron transfer, you can see this, can absorb 2 electrons and carry the electrons elsewhere. Now, plastoquinone is similar, only in chloroplasts. So this is animal and this is plant. And this is some sort of a sugar carrier. All right, guys, do you have any questions about the lipids? So we took us 3 lectures, which is good. I think we talked about a lot of interesting things. Hopefully you learned a lot about lipids. And we will also use, so we're not done with lipids yet. I just introduced all the main players in on the lipid arena. And then we're going to use those when we talk about membrane transport. So obviously these lipids are important constituents of cell membranes, and they play major roles in the function of cell membranes. So the homework for chapter 9 was due this morning, and there's the one for the lipids is due on Saturday. So don't forget about that. So you have 5 or 6 days to do that. And Wednesday we start in membrane transport. And I'll be working on some extra credit stuff for you this week. Sounds good? All right. Talk to you on Wednesday. Stay safe.
Thank you.